Neurons are cells within the nervous system that are responsible for receiving and sending information. Information from other cells is received through the dendrites, and messages are sent through the axon using action potential. When a neuron is at rest, not transmitting via action potential, it has a resting membrane potential of negative 65 megavolts. This potential is caused by more positively charged cations moving out of the cell than into the cell. When the neuron is at rest, more sodium ions are found outside the cell membrane, and more potassium ions are found inside the cell. These ions pass through passive ion channels in the membrane in order to establish an equilibrium of the concentration gradient, in which there are an equal number of sodium and potassium ions inside and outside of the cell. However, active sodium and potassium pumps use ATP created by the mitochondria to pump the ions back in and out of the cell, thus preventing the equal concentration gradient from occurring. Both cations and our negatively charged anions also attempt to go through the membrane in order to establish an equilibrium of the electrochemical gradient, a resting potential of 0 megavolts. However, the pumps again use energy in order to pump ions across the electrochemical gradient, thus maintaining the resting potential of negative 65 megavolts.
A neuron receives a signal from another cell through its dendrites, which triggers ligand-gated sodium channels to open and allow sodium ions into the cell. This causes the membrane potential of the cell to increase, known as the initial depolarization. If the potential reaches the threshold of negative 55 millivolts, an action potential is triggered.
When the membrane potential reaches the threshold of negative 55 megavolts, an action potential is triggered. Voltage-gated sodium channels open, allowing a rush of sodium ions into the cell that causes the membrane potential to spike to 35 megavolts. Once the potential reaches that point, the voltage-gated sodium channels close, and the potassium channels open, causing a rush of potassium ions to leave the cell and repolarize it. The cell's potential continues to decrease below its resting potential and be hyperpolarized. In this period, called the refractory period, the cell cannot have another action potential, forcing the signal to continue to travel down the axon and not go backwards. After the hyperpolarization period, sodium and potassium pumps use ATP to pump ions in and out of the cell and return it to its resting potential.
Action potentials move down unmyelinated axons by impacting one segment of the axon at a time. In a myelinated axon, action potentials move using saltatory conduction. Myelin sheaths act as electrical insulators for axons, allowing action potentials to jump quickly over large segments of the axon. Nodes of Ranvier are places between myelin sheaths which serve to generate fast electrical signals that are then carried by the myelin sheaths to the next node of Ranvier.
By understanding the process by which our mind communicates with our body, and vice versa, we can determine how motor function is impacted by certain neuronal diseases, and thus help us determine how best to treat them. Basic understanding of neuronal communication will be a building block for future research on how to improve motor function in humans, and hopefully will be used to find cures for diseases such as Alzheimer's, which disrupts neuronal communication.